next speaker is Dr. Eric Kandel. Eric did his undergraduate training at Harvard, his medical training at NYU Medical School in New York. He then became interested in psychiatry and neuroscience, went to the NIH and trained, went back to Harvard for training, and then joined the faculty at Columbia Medical School in New York as the founding director of a new brain research center and institute, which he still directs today. He's had a big donor, I think Kavli is the name of the individual who gave some money to that institute that Eric directs. Eric has always been interested in cell communication and signaling in the brain, and he's worked in a variety of models. He works with a sea slug called Aplysia. Doesn't have very many neurons, can't be very smart, but it does have memory, and it's been an ideal system that Eric's developed to study for various reflexes and memory of withdrawal reflexes and so forth. He also works with rodents, mice and rats as well. He has received many honorary degrees. He's in a number of societies, including the National Academy of Science. He's received the Lasker Award, the Gardner Award from Canada, the Wolf Prize from Israel, the Heineken Award from the Netherlands, and also the National Science Medal. So he has a number of honors, including the Nobel Prize, which he shared with two colleagues, independent scientists. Paul Greengard was one, also in New York at Rockefeller, for their work with neuroscience and cell signaling. Eric, welcome back to Houston again. Thank you for coming.
Thank you, Ferret, for those very generous words. I'm delighted to be here and to participate in the symposium for a number of reasons. First, it's hard for me to think of a theme that is more important in general and more meaningful to me personally. And I'm really delighted for Shelley Rubenfeld for organizing this. And second of all, it's an enormous privilege for me to talk back-to-back with Jim Watson. He not only made the most important biological discovery of the century, 20th century, but he's had a vision of neuroscience, a vision of biology that is quite extraordinary. It's fair to say that the unification in the biological sciences that occurred since 1950 has been one of the great intellectual treats of the last 55 years, and that the molecular biology that Jim pioneered is largely responsible for that. Very early on, he realized that molecular biology would also have an impact in the nervous system, and he's been an enormous support of me personally and of the field as a whole, and has made possible the unification of what we now call a new science of mind, the bringing together of psychology, neuroscience, and molecular biology with the hope of solving the problems that the psychiatrists have so far not been able to solve. I had the privilege of seeing Jim's slides before the talk, and he and I have talked about what we should present. And I'd like to sort of take a parallel path in terms of my own upbringing and my own work. So I've been interested in the problem of memory most of my mature life, and that interest came from my upbringing and from my circumstance as a Jew in Vienna. And I thought I'd begin by just telling you a little bit about what my life in Vienna was like, and then going on to tell you how that influenced my work afterwards. So I'm going to speak about my search for memory, beginning with Vienna. Then I'm going to speak a little bit about gene-environmental interaction and the persistence of memory storage. And then I'd like to focus on one issue that we have not yet addressed in thinking about the Holocaust, and that is the banality of people who commit evil acts. So let me begin with Vienna. I was born in Vienna, November 7th, 1929, to Hermann and Charlotte Kandel, my parents, who met in 1923 and married soon thereafter. And my father, as I will show you, ran a small toy store, and my mother worked with him, and we had a housekeeper who took care of us at home. My brother, Louis, and my brother is 5 years older than myself. He was born in 24, I was born in 29, had a fairly conventional, lower middle class upbringing. My parents were relatively poor. They did not have a great deal of money. And they ran this small toy store. I don't know whether you can see Hermann Kandel, 31 Kutschke Markt. For the longest time, I had the illusion that this was me. It actually, I think, more likely is my brother, and this is my mother. I'm not sure you can see that. This store still in existence. It's now a gourmet delicatessen. The woman actually is very pleased at the fact that my father had the store. Life in Vienna was moderately pleasant for Jews. They were very productive there and very successful there. But one was always aware of a certain degree of anti-Semitism, even before Hitler came. The Nazi Party, which was outlawed in 1934 in Austria, was quite active before that. And anti-Semitism was built into the fabric of Viennese society. Lueger, the mayor of Vienna in 1900 to about 1908, tutored Hitler. He made Hitler realize that anti-Semitism is an extremely effective political platform. He actually built it into the platform of his own political party. Even after Lueger and after Hitler had left, this is a protest in 1928 of the Nazi Party about an opera that is going to be performed in the Vienna Opera House, which is a great opera house, written by a Jew, actually involving a Negro. So the combination of Jew and Negro is enough to make it enormous protest, that this would lead to the degeneracy of the operatic life in Vienna if this was performed. But despite that, our life, I think, was reasonably peaceful, and I really remember my life in Vienna as being rather pleasant, unexciting, but pleasant. And then things changed dramatically. On March 13th, 1938, Hitler moved into Austria. One fully expected there would be significant opposition to Hitler's annexation of Austria. But to everyone's surprise, even his, because this is why he decided to go ahead with a formal annexation, he was welcomed enthusiastically by the crowds all over Austria where he went. He first arrived in Linz, and then he came to Vienna. This is the greeting in Vienna. He went to the Heldenplatz. This held several 100,000 people. They screamed enthusiastically, "Hail Hitler! Hail Hitler!" This is the hero that has unified the German-speaking people. My brother, who was very talented, had built a crystal radio set. And I will never forget listening to the Horst Wessel song, this martial music coming across as Hitler came into Vienna. It was absolutely frightening. We knew there was really no future for us. I experienced it immediately myself. The next day after Hitler came in, I walked along the outer ring. A classmate of mine walked up to me and said, "Kandel, my father said I'm never to talk to you again," and not a single person talked to me. Jews were beaten up terribly as soon as Hitler arrived in Vienna. They were asked to scrub the streets of Vienna, even with toothbrushes. My father was forced to scrub the streets. The streets had been marked with propaganda slogans because a plebiscite was going to be held between Schuschnigg, the Chancellor of Austria, and Hitler, whether there's going to be a unified country. There was every reason to believe that Austria would remain independent. And when Hitler sensed that, that's when he came across the border. And that's why everyone was surprised that he was received with such enormous enthusiasm. One capability the Austrians have is to change their mind. And within 24 hours, the same newspaper that opposed him said, "We now really welcome this great leader." The beating up of Jews in those days was one of the most vicious things that newspaper people who had covered Hitler in the 5 years he'd ruled in Germany commented on. A German dramatist called Zuckmayer, whom I later wrote about in my honors dissertation at Harvard, left Germany because he was too progressive. He knew that Hitler would be out to get him. He came to Vienna. He saw what happened, and he wrote in his autobiography the following: I've experienced some of the most horrible things of the 20th century. I fought in the First World War. I saw gassing up at front. I saw my comrades shot dead next to me. I saw the putsch, the Hitler putsch in Munich in 1923. I saw Hitler's rise to power in Berlin in 1933. Nothing, absolutely nothing, was comparable to what happened in Vienna in 1930 on March 13th. It was as if hell had opened up its doors and spewed out all the most horrible venom in the world. All the Jewish kids in my class were shunted out of the school within weeks. They were sent to a special school. We were beaten up when we went to the park. It was very, very difficult to walk along any street without being afraid that you were going to be beaten up. But this difficult situation became even worse on November 9th, 1938, which some of you may know as Kristallnacht. Just to give you a little historical background, on October 28th, Hitler moved a large number of Polish Jews living in Germany out of Germany and just dumped them on the other side of the border. A son of one of those people moved, Herschel Greenspan, was so unbelievably upset by this that on November 1st or 2nd, he was living in France, he marched into the German embassy in Paris and shot von Rath, an official, turns out a minor official, it's unimportant, of the German embassy, thinking he was the ambassador. In response to this act, Hitler destroyed every single synagogue in Germany and in Austria. Moreover, he moved lots of Jews out of their stores and out of their apartments. November 7th was my birthday. I'd gotten, as a birthday present, something I'd always wanted, a car from my father's store which you could drive by remote control, a blue car. I remember driving that with pleasure for 2 days. On November 9th, a knock on the door. 2 policemen came to the door and said, you've got 20 minutes, pack your belongings, you're going to another Jewish family, and we'll let you know when you can come back. We took a few things, spent a week with a Jewish family more affluent than we are, that we had never met before. We were extremely uncomfortable, so were they. And we were particularly worried because my father had not yet returned from work, and we did not see him for that whole week. After a week, we were allowed to return to our apartment. What we found bore no resemblance whatsoever to what we had left. Everything of value, and we didn't have that much of value, everything was gone. My mother's fur coat, several of my father's suits, some linen, small piece of jewelry, and my toy car. We had known before that we had to get out, and we got out as rapidly as we could, and we got out in stages. My grandparents left first. The reason we were able to come is because my grandparents' oldest son, my mother's brother, had emigrated to the United States in around 1928 and was established here and could send us affidavits to assure that he would support us. And so my grandparents came first. This was the quota requirement. Parents could come first. Young children could come second. My brother and I, so he was 14 and I was 9. We crossed the Atlantic by ourselves. I'm afraid to have my grandchildren go across the street by themselves. We crossed the Atlantic by ourselves, and my parents came last, just days before the war broke out. Now, it's hard to in any way reconstruct what happens in one's adult life based upon one's remembrances of childhood. But I've had the deep feeling that my interest in memory, my trying to understand what happened in Austria and Germany, has been one of the great motivating forces of my life. I wanted to understand how could people who listen to Haydn, Mozart, and Beethoven one day go out and kill Jews the next. The whole memory of that Viennese experience has never left me, and I've returned to it in a number of ways. And much of my scientific life has been devoted to trying to understand what the mechanisms of memory is in order to get some deeper biological insight into it. I came to the United States, and from the moment I arrived in Hoboken, I felt a freedom that I've never experienced in my life. This country has many flaws, but after you've lived in Vienna for 9 years, you think it's a pretty perfect country. My father used to call it the Golden Medina, the Golden State, because the opportunities are so fantastic here. And I remember we came, we did not have a single cent. My father sold women's clothing from door to door until he had enough money to open up a store. He didn't think he could go with a toy store here because at that time, actually, Americans weren't interested in elegant toys, mostly baseball mitts and footballs, which he wasn't familiar with. And with that, ultimately, he opened up a dry goods store, a haberdashery in Brooklyn, 411 Church Avenue, that supported my brother and myself through school. I went to a parochial school when I first arrived here, the Yeshiva Flatbush, and then I went to Erasmus Hall High School. And since Jack Byrne is in the audience, he won't believe me, I was co-captain with Ron Berman of the Erasmus Hall track team. While I was at Erasmus, as I was leaving, well, slightly before leaving, Mr. Campana, my history teacher, I had already become very much interested in history, trying to understand what happened in Austria and Germany, approached me and he said, Eric, where are you going to college? And I said, I'm going to Brooklyn College. And he said, why don't you apply to Harvard? And I said, well, I hadn't thought of it. Let me discuss it with my father. So I went and discussed it with my father. He said, look, we've already paid $10 for you to apply to Brooklyn College. We don't have a lot of money. So Brooklyn is fine. Your brother is going to it. Brooklyn is an excellent college, I must tell you. I went and I told it to Mr. Campana, and he said, here's the $10. Why don't you apply to Harvard? And I was accepted in Harvard, and my life was altered. I majored in history and literature, and I wrote my thesis on the response of intellectuals to National Socialism. I selected 3 German writers. One of them was Zuckmayer. And I was going to do graduate work in intellectual history, and then I fell in love. It happens to people. It happens to the best of people. I fell in love with a wonderful woman who is still a very good friend of mine, who was the daughter of 2 wonderful psychoanalysts in the Freud circle. And they got me extremely interested about psychoanalysis and Freud. Now you have to realize, most of you are too young to appreciate this, but in 1950, Freud was the exciting intellectual frontier. He gave one a completely new, rich, nuanced insight into the human mind, much more sophisticated than what Skinner had taught us or Watson had taught us. And I began to read actively about psychoanalysis, and I decided, my God, this is really interesting. If you want to understand why people do things, if you want to understand human motivation, you really have to understand the mind. Psychoanalysis is going to be the golden road to the mind. I had been close to finishing my junior year. I had not taken one science course at Harvard. I decided in the spur of the moment I would go to Harvard Summer School. I took an elementary chemistry course, and I was accepted medical school in the fall with the idea that I would fill all the requirements before I started the following September. And without going into details, I took 2 science courses my senior year, organic chemistry in the summer between my senior year and starting medical school, between graduating and starting. And I started medical school with the idea of becoming an analyst. At medical school, this friend and I broke up, and I had several other friendships, and then, I won't go into detail. I found myself in my senior year with an elective period coming up. And at NYU Medical School, we had a 6-month elective, and I decided, what should I do? By that time, I'd learned a little bit about DNA. There was a guy called Mark Adams who belonged to the phage group, and he was really so excited about what this new biology was going to do. So I thought, maybe I should learn something about the biology of the brain. Now, granted, that's not what most psychoanalysts were thinking about, but I should learn something. So this was a very small field. There was actually nobody at NYU who did anything on that. But I went to Columbia, where there's a guy called Harry Grunfuss, who was quite outstanding. We corresponded, he accepted me, and when I went to see him, he said, well, what is it that you want to work on? And I said, I want to find the anatomical substratum of the id, the ego, and the superego. And he looked at me as if I was Meshuggah. He looked at me as if I was out of my blooming mind, and he said, look, I don't even know whether those things exist. If they do, there's just no way we know how to go after those structures. The key insight to studying the brain is to study it one cell at a time, a reductionist approach, if you will, a continuation of what Jim and all modern biologists have been doing. And in his lab, I learned how to record from cells one at a time, and I worked with a number of preparations, recording from single cells, making microelectrodes. And at the end of my stay, he said, to my surprise, he said, you know, you're going to be drafted now. When I got out of medical school, 56 physicians were in the very plan. They were drafted, and they took care of soldiers and their dependents. But a few that were selected would be eligible for the NIH. And Grunfuss asked me, to my astonishment, because I had shown no talent whatsoever, would you be interested in going to the NIH? And I said, of course. So he recommended me to the head of the neurobiology laboratory, neurophysiology laboratory, Wade Marshall, and I was accepted simply on his recommendation. So once I got there, Wade was in a very interesting phase of his career. He had essentially become a full-time administrator, and he let people do pretty much what they wanted. And so I began to think of what I would like to do, and I decided memory storage. That is a wonderful problem. That is a key issue in psychoanalysis. You're constantly living through your memories. Conflict comes from repressed memories, and the whole idea of a therapeutic experience is to work through those earlier experiences in a trusting environment and to become more comfortable with them, to get command over them. So I thought that maybe I would use a cellular approach to memory storage. Brenda Milner had just the year before described a case of a young boy, HM, who had, for reasons of chronic epilepsy, removal of both sides of the temporal lobe. This is the temporal lobe here, and a structure deep to it called the hippocampus. And she found that those structures are critical for complex memory storage. And she particularly pointed to this deep structure here, the hippocampus. So I thought I would apply a cellular approach to the hippocampus. Now, the NIH, when I was there, 57 to 60, was nirvana. You could be a complete bozo, which I was. There were all kinds of bright people around you, and you can get their help. This is actually not just typical of the NIH, this is typical of American science. So I began to speak to people about applying cellular techniques to the hippocampus. No one had done this before, and they said, this is extremely hard, unlikely you'll succeed, but if you want to do it, go ahead. So I started to do that, and I was very lucky. A guy called Alden Spencer, we used to call each other draft dodgers, he also joined the lab, and I talked him into this project because I realized it would be difficult, and he turned out to be a spectacular collaborator. He was a lifelong friend of mine. And he and I succeeded within a few months of obtaining the first intracellular recordings from the hippocampus. It was really a major event. Two incompetents had made a significant step forward. Wade Marshall was ecstatic. He was running up and down saying, look what Eric and Alden have done. I gave a major talk on this. not quite as large an audience as this, but let's say, a tenth of this audience was there. And people were very excited. And Ald and I were very excited just for the flattery of it all. And then we realized, what the hell had we learned about memory? Not a damn thing. We learned how cells in the hippocampus function. And only then, we were so dumb, we were so illiterate, did we begin to realize that, memory involves alterations in information coming into a structure. Learning modifies the information coming in and then stores it in memory. You've got to look at what's coming in and how that's altered. You've got to look at what we call the synaptic connections coming in. So we started to see what is the nature of the input coming into the hippocampus. And we used light stimuli and tactile stimuli and sound, and we got nowhere. Turns out it's very complicated what's represented in the hippocampus. It's multidimensional, it's space. It's taken a long time to figure out what's going on. So I began to sort of look around for a simple preparation in which to do this. I said to myself, look, the hippocampus is too complicated. I've got to take a radical reductionist approach. Rather than looking at the most complicated form of memory, I want to look at the simplest form of memory. I want to look at in the simplest animal possible. Let me try to understand the most elementary memory storage and see how it works. By that time, I had married, and I married a French woman. And one of the reasons I married her is because when I was making this difficult decision, I discussed it with her. And I said, Denise, I'm not sure I should go into neurobiology. That is, decision to go to the NIH and go to neurobiology, because I don't have any money and you don't have any money, and one of us has to earn something. And she said, absurd. Money is of no significance to me. She has never repeated those sweet words again, but at the time she said it, I was just blown away. So when I began to look around for different systems, I talked to her about this, and we discussed the advantages of crayfish and leeches and lobsters, and I ultimately focused in on a marine snail Aplysia I'm going to show you in a minute. And there were only 3 labs in the world working on it. I'm sorry, 2 labs in the world working on it. Both were French. My wife is French, is a good thing. We had them both for dinner, very nice people. One was a woman, Angelique Avanitaki Charlesonitis, and the other was Ladislav Tork. Charlesonitis lived in Marseille. Tork was in Paris. Denise is a Parisienne. She said, Marseille? That's like going to Albany. We're going to Paris. So I went to Paris, and I started to work in Aplysia. Now, why would you want to work in Aplysia? This is your brain and mind. It's got lots of neurons, millions of neurons. Aplysia has a very simple nervous system. It has 20,000 nerve cells. These are collected in clusters. There are about 10 of them. Each has about 2,000 nerve cells. So there are few cells. In addition, the cells are absolutely gigantic. They're the largest nerve cells in the animal system. They're the largest nerve cells in any animal. Moreover, the cells are so distinctive in their pigmentation, their coloration, position that you can identify many of the cells as unique individuals and give them names like Jack and Susan and Jim and Ferret and return to the same cell. Imagine returning to Jim Watson every single day you wanted to, the same cell right there. So this obviously made it much easier to work out the neural circuitry and for any behavior, so I began to focus in on that. And in the simple animal, with the simple nervous system, I focused on the simplest behavior the animal had. This is the head of the animal, this is the tail of the animal. It has a respiratory organ called the gill that is normally protected by a sheet of skin called the mantle shelf that ends in a fleshy spout called the siphon. If you apply a weak stimulus to the siphon, you get a withdrawal of both the siphon and the gill, seeking protection. So it's like the withdrawal of a hand from a hot object. Now, this simple reflex withdrawal, amazingly enough, can be modified not simply by one, but by several different forms of learning. All of them very simple: habituation, sensitization, classical conditioning. Recently, Jack has shown operant conditioning. Moreover, each of these forms gives rise to a short-term form and a long-term form. So the first thing we learn is even simple animals, in fact, even the simplest reflexes in simple animals can be modified not simply by one, but by several different forms of learning, and each of them can give rise to a short-term and a long-term form. We then took advantage of the very simple nervous system that the animal has and these large identified cells in order to work out the neural circuit of the behavior. And Jack Byrne, who's in this audience, who was my first graduate student, had a key role in this. We found there were precisely 24 sensory neurons, Jack found that made precise connections to 6 motor neurons and connections to excitatory and inhibitory interneurons. These connections were invariant. In every animal, the same cells connected to exactly the same follower cells. This was quite nice, and it's turned out to be a general rule. There's enormous precision in the wiring of specific behaviors, but it raised a paradox. How do you get so many different forms of learning if the behavior is so rigidly specified? So to explore that, we actually looked at a form of learning called sensitization. Sensitization is an enhancement of reflex response when you give the animal a scary stimulus to the tail. So look what happens. When you stimulate the tail, you activate a learning system, a modulatory system that uses serotonin as its transmitters. It acts on the sensory neurons, including on its presynaptic terminals, and strengthens the connections between the sensory neurons and the motor neurons, and between the sensory neurons and the interneurons. If you stimulate the tail once, you produce a memory that lasts minutes. It's called short-term memory. That produces a functional change in these connections. They change for a period of minutes, but there's no anatomical change. But if you stimulate the tail repeatedly, minimum 5 times, you produce a long-term memory that persists for days, and that's associated with a growth of new connections. So long-term memory involves an actual growth of synaptic connections. So if you remember anything about this lecture, you're more likely to remember Jim's lecture. You will come out of this lecture with a different head than you walked into. Now, hold on to that idea because I'm going to come back to it later on. By this time, molecular biology had come along. I was very fortunate to collaborate with a guy called Jim Schwartz, who was a molecular biologist. And we began to explore what happens on the molecular level. We found that if you stimulate the tail, you activate these serotonergic cells, and they activate a second messenger cascade, a signaling system within the cell. And that signaling system gives you a transient strengthening of synaptic connections between the sensory neuron and the motor neuron. But when you give repeated stimulation, the signaling system moves into the nucleus. And there it activates genes. And it activates 2 kinds of activators of genes. One is a protein that turns genes on, and another one is a protein that turns genes off. So one of the things you've got to do is to remove an inhibitory constraint in order to activate genes that are important for long-term memory. Once you activate the Krebs activator, it acts on a whole bunch of downstream genes. I've simplified this for you to give rise to the growth of new synaptic connections. So there are several interesting things here that I would like to point out to you. The first is that you have an inhibitory constraint. Isn't that odd? We would think you'd like to put everything into long-term memory. Why would you want to have an inhibitory constraint on it? Why would you want to make it difficult to put things into long-term memory? And if you just think about it, you realize that many things that happen to you are miserable. Many lectures that you hear are boring. You want to forget them as soon as possible. Something has to be really important for you to put into long-term memory, number one. Number two, once you remove the inhibitory constraint, you activate Krebs, and this acts on downstream genes. And I should tell you now, this is quite a general pattern. While we were working on this, people in Jim's lab, Tim Tully was seeing this in the fly as well. We now have seen this in the mouse. It activates genes in order to give rise to the growth of new connections. So when you think of genes normally, that is the person in the street, they think of genes as being the controllers of behavior. They may not be aware of the fact that certain genes, particularly genes in the brain, can be regulated by environmental experiences. So again, if you remember something, it is because gene expression in your brain is altered, and that's a requirement for the growth of new synaptic connections. Once CREB is activated, this growth occurs. Let me just show you what the growth occurs. In your brain, it is not this dramatic. But this is a single sensory neuron of Aplysia, a single motor neuron, before and after training for this form of learning, sensitization. There is a sprouting of new processes, both from the presynaptic neuron and the postsynaptic neuron, sensory neuron and the motor neuron. There's a doubling number of synaptic connections. Now you're going to say, this is a lousy snail. Does this apply to you and does it apply to me? This is you and me. The gentleman in the audience, I should tell you, this is a pre-Victorian view of the homunculus in the brain, the genitalia adequately represented. This is what you look like to yourself when no one else is looking. This is the body surface. Your skin is represented in a systematic way, just like I showed you the gill withdrawal reflex, in a systematic way on the surface of the brain. There's a specific area that represents the brain, and it is not a literal translation. Those parts of the body that are more important, like the fingertips, for tactile sensation, have a larger representation. Now, when I was a medical student, I was taught that that representation is fixed. We now know that this is not true. Mike Merzenich examined 6 different monkeys and found that each one had a somewhat different hand representation. Each finger in the hand has its own representation, a little strip devoted to it. So he had monkeys, in order to ask, is this genetically determined or is this environmental? Is this due to learning? He had monkeys press a bar for food for several weeks, and he found that the fingertips that the monkeys used to press for food, that representation expanded in the brain as a result of that usage. This has now been found in people. It's been found in a variety of different circumstances. Moreover, as your mother told you, if you start doing this early, you'll have a greater enhancement than if you started later on. This is a study of Traub, of musicians, playing the fiddle. These are string instrument players. The string instrument players use their right hand to bow. There's nothing to bowing, yet your hand acts like a fist. The fingering is done by the left hand. So if you look at the distribution of the right hand in string instrument players and normal people, non-instrument players, there's no difference. But the left hand, there's a dramatic difference between string instrument players and people who don't play string instruments. And if you do what your mother told you, you start early, before puberty, the representation is larger than if you start later. In 1990, Kopecky and Smithies developed techniques for knocking out genes in mice, and that made it obvious to my colleagues and myself and to many other people that mice would be terrific systems to apply a molecular approach to study learning and memory. And we began to do that at Suzuma Tanagawa and lots of other people, and we now have a much better insight into that very structure that I was struggling with at the beginning, the hippocampus. We understand more of how it works, and we are beginning to understand how long-term memory is mediated by it. And just to give you a very simplified view, this is the hippocampus, this is the Aplysia, this is lots of people's work, I'm just summarizing it for you. But even though there are enormous differences, the neural circuitry is more complicated, many things are more complicated, the storage mechanism shares a number of features in common. There's a CREB activator, there's absolutely critical, there's a CREB repressor, and there is increasing evidence for the growth of new synaptic connections. So as a result of this, I was fortunate to receive a telephone call on Yom Kippur, the year 2000, be informed that I have won the Nobel Prize together with Arvid Carlson and Paul Ringard. I will never forget that Jim Watson called me soon thereafter to congratulate me, threw a very nice party for me at Cold Spring Harbor, and he said, "This is the happiest day of your life. Remember, be gracious to other people." Now, he knew what he's talking to because I'm not a person who's very gracious, so I needed that advice. This is my granddaughter, Allison. She doesn't wear glasses. She has contact lenses now. And this is my granddaughter, Libby. Jack Byrne, who's sitting right there, really felt that the Nobel Prize should have gone to Aplysia, and he sent me this picture, which I use in my Nobel Prize speech. And it brought down the house and made me feel very relaxed. How does that bear to the theme that we began with? And I'd like to conclude with just a few sort of free associations on that. When I won the Nobel Prize, you get lots of telephone calls. Girls that had rejected me earlier all of a sudden called me up and invited me for a weekend in the country. And lots of telephone calls came from Vienna. How wonderful to have another Austrian Nobel Prize. We're so proud of you. And I told them, look, Bubby, you got this wrong. This is an American Nobel Prize, an American Jewish Nobel Prize. So the president of Austria wrote me a letter, and he said, what can we do to recognize you? And I said, Mr. President, I have more recognition than I deserve. What I'm struggling with is the difficulty that Austria has is confronting its past. I would like to see a symposium at the University of Vienna that concerns itself with the response of Austria to National Socialism. Claysdale was fantastic, so I asked Fritz Stern, who's a historian, to help me, and we organized a symposium in Vienna. And one of the things that has really changed, somebody asked me today, how does Nobel Prize change your life? I mean, you can see with Jim just has to say anything and all the newspapers pick it up. Newspapers don't pay much attention to me in the United States, but anytime I say anything in Vienna, it's picked up. So I said, what I want to understand is, how can people that love Goethe and read Schiller and recite this poetry in class be so horrible? What accounts for this banality of evil? And people have thought about this, and Robert Lifton is one of the people who's thought more extensively about this, has argued that there are sort of three kinds of people that get involved in this transformation: ideologues, opportunists, and fellow travelers. I'd like to say a word about each of them. Let me begin with ideologues. This is a very famous picture. This is the dean of the medical school of the University of Vienna, Edward Pernkopf, an ideologue. He greets the medical faculty of the University of Vienna. This is less than a month, so this is 3 weeks after Hitler came in. Hitler came in March 13th, April. And he himself, I'll show you a bulb of the picture. I don't know whether you can see it. He's in an SI uniform, a brown uniform. Here is a portrait of Hitler in the back. The faculty are all giving the Hitler salute. Now, why are the faculty giving a Hitler salute? 50% of the faculty was Jewish. This faculty was fired. What remained was the remaining faculty. These now saw a wonderful opportunity to move ahead. So what Hitler created, and this has been repeatedly documented in Vienna, is what Jim referred to. Tremendous envy and opportunism. People saw an opportunity to really move ahead. Pankow himself was an ideologue, probably an evil ideologue. Not all ideologues need to be evil. Oliver Wendell Holmes clearly was not an evil man. People can be misguided or may get inappropriate feedback on what they think are virtuous ideas. Perkoff, I have no reason to believe is virtuous. I don't know whether the physicians in the audience remember the Perkoff Atlas. There was a very famous atlas. Only within 10 or 15 years has it been realized that that autopsy material that he used was based upon concentration camp victims that he dissected. So we have here examples of both ideologues and opportunists, and I just want to quickly take you through a number of these things. The book that was referred to earlier, the textbook in genetics, The Foundation of Human Genetics and Racial Hygienes, written by 4 famous people, Bauer, Fischer, and Lenz. Lenz was an extremely influential guy on Hitler, emphasized that the foundation of human genes and racial hygienes began to speak of eugenic measures. Fischer, one of the co-authors of this, was a director of a Kaiser Wilhelm Institute. Being a director of the Kaiser Wilhelm Institute is like being a director of a Max Planck Institute. It's like being head of a Rockefeller Institute. This guy was not only advancing euthanasia, but early on was one of the early members of the Nazi Party. Psychiatrists, my discipline, were particularly active in this. They were beginning to sterilize mentally retarded and schizophrenic patients before Hitler came to power. They were not only sterilizing schizophrenic patients because the evidence was already accumulating that it was a genetic disease, they were thinking of sterilizing first-degree relatives. So this idea that physicians could be involved in these kinds of practices preceded Hitler. Hitler took this book with him to prison when he was in prison in 1924, and it made a major impact on his thinking and on his platform. Psychiatrists, similarly, early members of the Nazi Party. Paul Nietzsche was one of the leading psychiatrists there, and later, as with other physicians, had a major role in the euthanasia effort. What is really remarkable, and what has been emphasized in the earlier talks, but is just amazing to me, is to what degree Hitler adopted this scientific policy. In fact, Lenz, who was one of the authors of this eugenics textbook, raved about Hitler, saying Hitler is the only leader in Europe who understands genetics. And many people said to Lifton, when he examined the perpetrators, the physicians, they said, you know, one of the reasons Hitler was so appealing to us is it was also scientific. You know, we were thinking along some of these lines before, but he came along, he laid out the whole scientific basis of it, and he made it interesting for us. So the amazing thing about the Nazi Party is it saw itself as, if you will, translational research. It saw itself having a scientific basis and appealed to many physicians because of that reason. So this is, Shelley referred to this earlier, this is the document that Hitler signed, which allowed Philip Bühler and Karl Brandt to set up these T4 facilities, which were the early euthanasia facilities. Karl Brandt is an extremely interesting guy. Karl Brandt is one of Hitler's personal physicians, an extremely intelligent, brilliantly trained guy who took upon himself the task of selecting the way euthanasia would be carried out, the way massive killing would be carried out. And he was the one that selected carbon monoxide as the gas to be, first of all, that gas should be used, and specifically carbon monoxide should be used for the killings. Very few of the physicians were ultimately brought to trial. He was one of the 23 physicians brought to trial, and he was sentenced to death. So you could understand that there are ideologues. Some of these are probably evil. Many have misled ideals. A second group are people who see this as a fantastic opportunity to advance themselves and join the cause because it's advantageous for them. But a number of people were just fellow travelers. Several people in the early days at Auschwitz complained about the fact that they didn't want to do this kind of work. These were the people involved in taking care of the inmates at Auschwitz. And it was explained to them that this is your duty. You didn't select these people. You don't have nothing to do with this. You just do your job and do it as well as possible. And these people were swept up in this and were willing to repress the horrors they were doing and focus on the task. And on weekends, they could go along and party and have a very good time as if life was going on, as if the rest of the time they were doing nothing when what they were actually doing was killing people. So I think this is really very interesting from both ultimately understanding it from a genetic and from an environmental point of view. It is my guess, and this is just absolute wild guess, that there probably will be sets of genes that predispose people to criminal behavior. And probably some of the things we saw, the Eichmanns, the Hitlers, the Goerings, the Goebbels, they may have had genes that predisposed them. A large number of people were banal. There were people who were perfectly reasonable intelligence, reasonable judgment in many areas, who somehow fell into this mode of thinking for one reason or another. They were socialized in it, either because it was advantageous or they were caught up with the ideology secondarily. And third, there were people who go along because they were intimidated, they were scared, and they talked themselves, or other people allowed them to talk themselves out of any personal responsibility for it. It is amazing, when Lifton interviewed the perpetrators, the physicians after the war, practically none of them felt any guilt. They all said, we were acting on a virtuous cause. Hitler defined this as a serious disease of society. We were physicians, our function is to treat the disease. If cutting out that offending group is what cures the race, that's what we're going to do. So, what does the biology that we have learned so far tell us about this? What does it tell us about environmental interaction? What does it tell us about the banality of people who commit evil acts? Well, to be honest about it, practically nothing. But I want to leave you with two thoughts. One is that I told you before that the learning values, the learning of evil action, is a learning process. It involves alterations of gene expression. And we know, for example, that as a result of the fact that we learn different things, every person in this audience has a somewhat different brain than every other person. Identical twins with identical genes will have somewhat different brains. And you can see how a dogma coming along, an ideology that sweeps people along, can produce long-lasting changes in people's minds by producing alteration in gene expression and growth of new synaptic connections. So one can see how these dogmas can really affect a society. Moreover, I just want to make one obvious point to you, and that is that you can get mental illness, and this is a form of mental illness, if you will, in one of two ways. One is by a mutation in the gene, in which you actually have an alteration. For example, in the DNA sequence, you have an abnormal messenger RNA, you produce an abnormal protein. And several of these combine together to give you schizophrenia and depression, so we now think. But in addition, you can also get a disease, a mental illness, without having any alteration in gene expression by simply turning certain genes on as a result of a traumatic event, for example, post-traumatic stress disorder, that were previously inactive, or turning it on to a higher level. So you can induce gene expression in a traumatic way that can also give rise to a phenotype. It will not be as severe as the genetically induced illness, but you can produce mental aberrations through environmental contingencies. So I simply want to point out that there are two ways you can produce alterations in gene expression. And let me just end up with three sort of philosophical notes, if you will. These considerations remind me of something that Reinhold Niebuhr said, of which I'm very fond. The capability of people to do good makes democracy desirable. Their capability for evil makes democracy necessary. And I think this is so absolutely right. The reason one needs a pluralistic society, a separation of power, is because one needs check and balances. The reason eugenics never moved further in the United States is not that the eugenics movement wasn't strong, but there were countervailing trends, and these did not exist in Germany. And the other thing is, and this is why we value the Jim Watsons of this world, people who speak up under all circumstances, people who are not worried about being politically correct. Eternal vigilance is the price of a free and independent society. Thank you very much.